Cell division. Prior to cell division, a cell will spend about 90% of its time in a phase known as interphase. So in this example, we are considering a cell that contains 6 chromosomes, so its diploid number is 6. The 6 chromosomes are inside the nuclear membrane, and we've got the nucleolus. Towards the end of the interphase, the DNA replication occurs by semi-conservative replication, and our number of chromosomes then double up. Now, when we look at these, we can see that our chromosomes are attached to each other at what's known as the centromere, and the parts, each one is known as a sister chromatid. So they're attached together at the centromere. At the end of interphase, we then enter into the stages of mitosis. So the first stage of mitosis is called prophase, and during prophase, centrioles move to the poles of the cell and start to produce spindle fibre. The nuclear membrane will start to break down as well, and the nucleolus will also disappear. So at the end of prophase, we have spindle formed, we have no nuclear membrane, and we have no nucleolus. Then we enter into metaphase, and in metaphase our chromosomes with the sister chromatids attached move to the equator or the middle of the cell. So I think of meta and met in the middle. So they move to the middle of the cell, and the centromeres are attached to the spindle. So the spindle attaches to each centromere during metaphase. Then we enter into anaphase. And during anaphase, this is when the spindle fibres contract and pull the sister chromatids apart at the centromere. So they pull them apart so that the sister chromatids move to the poles of the cell. Then it enters into telophase or telophase. And during this phase, the spindle fibre will disappear or break down. A nuclear membrane forms around each set of chromosomes, and you can see we have gone back to our diploid number that we started with of 6, 5, 6, and our nucleolus will reform. So at the end of telophase, we have 2 nuclear membranes surrounding our 6 chromosomes, our nucleolus, and then at that stage we enter into the whole cell dividing into what's known as cytokinesis. In an animal cell, the cell membrane will invaginate and break off to form our 2 daughter cells. In a plant cell, a cell plate will form, which is the precursor of the cell wall, and it will form down the middle. The Golgi bodies will produce vesicles which will gather at the middle of it and form this cell plate, and eventually it will surround and form the middle lamella of the cell and break into our 2 daughter cells. Cellulose will then form on each side of the middle lamella to form the cell walls of the 2 new plant cells. So our stages of mitosis are prophase, metaphase, anaphase, telophase.